Hello, and welcome to this Nature.com custom webcast titled Single-Cell DNA Analysis, Resolving Tumor Heterogeneity with Implications for Dynamic Therapy. My name is Joshan Karpan, and I will be your moderator. Today's webcast is sponsored by Mission Bio, and will be given by three speakers: Dr. Catherine Smith of the University of California, San Francisco, Dr. Alexander Perl of the University of Pennsylvania, and Dr. Dennis Eastburn of Mission Bio. Today's webcast will be covering the present state of clinical genomics and its applications and limitations, the need for single-cell sequencing to resolve bulk NGS challenges, their work together on the CHRYSALIS study using single-cell DNA analysis to determine tumour response, and finally they'll describe work with a novel single-cell DNA analysis platform, its capabilities and implications for dynamic therapy. We will then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Catherine.
Thank you for the introduction. So we'll talk first about the state of clinical sequencing using next generation sequencing platforms. Many large cancer centers have adopted next generation sequencing assays for clinical use. And there are also multiple commercial assays that are available through reference laboratories. These panels are used for both diagnosis, for prognosis, and also for treatment decision-making, or quote-unquote precision medicine, in which the idea is to potentially identify targetable lesions that may point toward potential pathways that could then be targeted by either small molecule or antibody therapy or others. Currently, these panels are DNA-based targeted panels, and they can be used either for solid tumor or hematologic malignancies or both. However, these panels differ widely. They differ in terms of gene composition and coverage. For example, some panels cover only hotspots, other panels cover all exons of the gene. They also differ in terms of capture method and whether they use germline controls. And in fact, most panels perform tumor-only sequencing. The other thing that's quite variable amongst different centers is the clinical report content, whether it includes variant allele frequency and what information, if any, is provided about the particular lesions identified. So two different capture methods can be used in these targeted sequencing panels. The first of these are either amplicon-based or PCR-based capture, and the second is hybridization-based capture, in which probes are customized to capture targeted parts of the genome, and they are subsequently amplified and sequenced. There are advantages and disadvantages to each of these capture methods. The amplicon-based method is both flexible and scalable, meaning that different amplicons and therefore different targets can be added or subtracted at any given time. However, given that it's a PCR-based method, it's also vulnerable to allele dropout, and it's also vulnerable to genes that are difficult to PCR, such as areas of the genome with high GC content or repetitive sequences. The issues that go with PCR are also problematic for amplicon-based panels, such as mutations that might be contained in the primer itself, the fact that large indels may be difficult to detect because they are less amenable to PCR amplification, and there can also be poor quality sequencing at the amplicon ends. In terms of hybridization capture, these panels are usually fixed, identified, and designed from the get-go and difficult to edit once designed. However, given that they're non-based on PCR, they're less vulnerable to allele dropout. And the capture allows interrogation of neighboring regions. However, on the flip side, this also means that there may be decreased coverage of the target regions. Using this method, however, it's easier to eliminate PCR duplicates, although the capture is sometimes sensitive to base composition, so not all regions of the genome are uniformly captured. Now, in most of these platforms, the variant allele frequency of the mutation in question is given. And this is defined as the fraction of mutant alleles in a given sample. For example, a 50% variant allele frequency is usually assumed to represent a heterozygous mutation in a population that is 100% tumor. However, this assumption is flawed for several reasons. Clearly, clinical specimens are not usually composed of 100% tumor cells. Also, there's clonal heterogeneity that exists in tumor samples, and also not all samples are diploid. Many tumor samples are aneuploid. There's also an issue of sensitivity, and most of these targeted genomic assays will have difficulty detecting variant allele frequencies of less than 2%, and most clinical sequencing platforms will therefore not report mutations that occur at a VAF of less than 2%. This slide illustrates some of the issues that are problematic about assumptions regarding variant allele frequency. For example, as you can see in the top of this slide, in a given population of cells where a mutation has a variant allele frequency of 33%, this can represent multiple scenarios. It can actually represent a scenario in which there is 50% of a wild type allele and 50% of a heterozygous mutant allele and 0% of a homozygous mutant allele. And that would give a variant allele frequency of 33%. In a second scenario, the wild type population could be 66% of the cellular population, and then 18% of the population is heterozygous and 16% is homozygous mutant, and that would also give you a variant allele frequency of 33%. And then in the third scenario, you could have approximately equal representation of wild type and heterozygous mutant alleles at 46 and 48%, and a small amount of a homozygous mutant allele at 6%, and that would also give you a variant allele frequency of 33%. And there's also a problem when you have multiple mutations within a given population. You can have multiple different genotypes for two different mutations. You could have homozygous A and heterozygous A, homozygous B and heterozygous B, and various different combinations. And bulk sequencing is unable to resolve these simply by looking at variant allele frequency. However, this makes it very difficult to estimate clonal composition. So there have been multiple papers that have been published that have attempted to estimate the clonal population of a given tumor population from variant allele frequency. This is done usually by inferring from the variant allele frequency and estimating the clonal composition of the tumor sample. However, these estimates are flawed because they're reliant on sequencing coverage and depth, and low frequency mutations are often not detected, especially when you have low sequencing coverage. And if low frequency mutations are not detected, then low frequency clones may also be missed. Also, there's often in studies in which the idea is to look at clonal evolution that occurs with treatment over time, you may be able to postulate clonal evolution from changes in variant allele frequency from bulk sequencing. However, there are multiple scenarios that are often possible from these estimates, and so therefore these become guesses rather than true observations of changes in the actual tumor clones that occur over time. These issues can actually be illustrated in this study, which was done in acute myeloid leukemia. On the left, you see the estimate of clonal selection that occurs with low coverage sequencing. And you can see that initially, at the time of diagnosis, there were 3 cancer clones that were detected, and there was subsequent selection for a red and gray clones after chemotherapy that ended up becoming the bulk of the relapsed population. However, on the right, what you see is the results of the clonal evolution that's estimated with high sequencing coverage, which revealed multiple other low-frequency clones that were not detectable using standard next-generation sequencing coverage. So to talk more about issues of clonal heterogeneity and acute myeloid leukemia, I'm going to pass it off to my distinguished colleague, Alexander Perl.
Thanks, Kathy. I'm going to talk about genomics and heterogeneity of AML, and I thought I would start with an overview of normal hematopoiesis and leukemogenesis, just to orient the watchers of the program. Normally, we think of the bone marrow as being a place where there's dramatic heterogeneity of cell morphology and polyclonal hematopoiesis arising from up to tens of thousands of hematopoietic stem cells. And what we see under the microscope is what's shown here in the schematic, which is there's relatively few immature cells that are mitotically active, and these are shown as myeloblasts, which can account for just a few percent of the bone marrow, but the vast majority, more than 95%, are postmitotic cells that have a range of morphologic features from the most immature promyelocytes to the most mature cells called neutrophils, and everything in between. And under normal circumstances, these cells that are differentiating or undergoing maturation are the vast majority of what's seen in the bone marrow. And the proliferative fraction is really quite small. So again, normally there's just maybe 1 or 2% myeloblasts, and a vast majority of the bone marrow is this large number of differentiating cells. Now what happens in acute myeloid leukemia is exactly the opposite. What we see in the bone marrow is that the mature cells are dramatically decreased, and in their place there's an expansion of the most immature cells that are usually arrested in differentiation when they're still at a proliferative phase. And under the microscope they look like myeloblasts. These are often 20, 30, 40, 50, sometimes 70, 80, or 90% of the bone marrow aspirate. And they take up the vast majority of the space in the bone marrow, crowding out the ability to make normal blood cells. The transition from normal blood-making ability to the expansion of the leukemia cells happens rapidly, usually over maybe just a few weeks. So patients go from well to sick very quickly, and it depletes them of their normal blood cells numbers in the blood. So we see a dramatic drop in their white blood cell count, their red blood cell count, and their platelet count for the normal mature cells. And in their place, we often see an increase in these leukemic blasts in the blood as well. So we can see the leukemia cells, which are clonal, both in the bone marrow and also in the peripheral blood. And they are really the same cells. And again, they are mitotically active. If you look here at this bone marrow aspirate of a normal bone marrow on the left, you can see that there are relatively few myeloblasts. It may be a little bit hard to see if you're not a hematopathologist or a hematologist, but if you take my word for it, the number of myeloblasts in the picture on the left is really only about 1%, and that's normal. It contrasts with a marrow aspirate from a patient with acute myeloid leukemia shown on the right, where nearly all of the cells look the same, and again they have the features of mitotically active cells with nucleoli, relatively minimal cytoplasm. These are the features of myeloblasts, and this is what we see in AML. And we see similar features in the peripheral blood too, with an increase in the myeloblasts and a decrease in the number of normal white blood cells, red blood cells, and platelets. So where does this come from? As I mentioned, the leukemia cells are clonal proliferations and contain recurrent mutations. Where do those mutations come from? Most commonly, they occur as part of the aging process. And as we get older, our bone marrows get older as well. And we know that the stem cells that are in our bone marrow were there prior to birth and have to last our entire lives. And they're subject to acquisition of mutations just through the aging process. They divide very rarely, so the acquisition of mutations in these cells is pretty uncommon. But the bone marrow is also one of the most rapidly dividing parts of the body throughout life, and so we have to turn over a huge number of blood cells every day. And so the acquisition of mutations is all but inevitable across the age spectrum. It just only rarely happens in coding regions that are important for hematopoiesis. But we do see it actually with some regularity. And if we sample the blood from otherwise healthy individuals, we actually see that as a function of age, there's an incidence of clonal hematopoiesis that can be determined by next-generation sequencing of a large number of genes. This is quite rare in young people, and the incidence increases with age, and actually represents upwards of 10% if we look at individuals aged 60 to 70 and even older. Why does this happen? It happens because mutations occur in hematopoietic stem cells that actually initiate clonal expansion of those cells, and a greater proportion of the blood is taken over by that stem cell, which leaves more cells that originated with that mutation to acquire additional mutations that might cause disease phenotype. Clonal hematopoiesis of indeterminate prognosis is called that because only a minority of people who have clonal hematopoiesis go on to anything wrong with the blood that either causes symptoms or really is considered a disease. One such condition is called myelodysplastic syndrome, and AML is yet another. And we know that there are people who likely start out with clonal hematopoiesis and then progress to MDS and then progress to AML. And similarly, there are people who have clonal hematopoiesis and then progress just to AML. The reason for this is likely dependent on which mutations are acquired and potentially also the order in which they are acquired. If we look at the incidence of AML and the incidence of clonal hematopoiesis, they're actually very similar. Those are shown on the two right figures. And the mutations that are affected in CHIP and in AML are often quite overlapping. So we think of these two processes as quite related. These are data from the Cancer Genome Atlas group, which was the first large-scale sequencing effort to look at the genomic landscape of cancer. 200 AML genotypes are shown on the right of the figure, with all mutations from coding and regulatory regions shown from individual cases. And as is summarized on the left, the AML cases had very few mutations per tumor. On average, there really were only about a handful of genes, 4 or 5 per case, that were recurrently mutated, and overall less than a dozen sequence alterations per tumor. And this contrasts with a number of solid malignancies, such as breast or ovarian, colorectal, or lung cancer, which have far higher mutation burden. And for this reason, each mutation appears to carry greater weight in terms of oncogenesis, and it's far easier to examine clonal architecture in AML than for many other kinds of tumors. And another advantage of studying AML is that you can repeatedly sample the tumor itself because the diseased cells are circulating in the bloodstream, and repeated sampling of the marrow is routinely performed during therapy. So while it's not unique in the ability to look at clonal architecture, in some ways it's a really ideal disease to study this question. If we look at the mutations that make up AML, the very most commonly mutated gene is FLT3, which is important diagnostically, prognostically, and therapeutically. Diagnostically, FLT3 is an oncogene that's recurrently mutated in leukemia, and particularly AML. It's not mutated really in other cancers. It doesn't show up in lymphomas, breast cancers, lung cancers, etc. The normal function of FLT3 is a hematopoietic receptor tyrosine kinase, and its expression in normal tissues is largely restricted to early hematopoietic cells such as HSCs and early myeloid and lymphoid progenitors. Now, in AML, certain mutations of FLT3 are prognostically important, such as internal tandem duplications, and these are associated with high relapse rates and poor survival. The function of these mutations are to activate the kinase, which I'll show on the next slide. Because these are activating FLT3 mutations, there's obviously been interest in developing drugs that inhibit the tyrosine kinase activity of FLT3. So that function is shown in a highly simplistic cartoon here, where we see that FLT3 exists with an extracellular ligand binding domain, and on the intracellular side, there is a split kinase domain or an interrupted kinase domain. The juxtamembrane region of FLT3 can be mutated in the form of insertions of variable size of FLT3 sequence that form internal tandem duplications, or ITD mutations. A second kind of mutation occurs as point mutations in the activation loop of the tyrosine kinase domain. These are most commonly at codon D835. The effect of either of these mutations is to favor the activated state of the receptor, and that turns on downstream signaling through a number of targets. These can be shown experimentally to involve phosphorylation of STAT5, signaling through PI3 kinase and AK2, as well as the RAS MAP kinase signaling pathway. Functionally, this leads to inhibition of apoptosis, enhanced proliferation, and differentiation block. So the first FLT3 inhibitors entered the clinic in about the year 2002, and they've gotten better over the years at getting both more potent and also more selective for FLT3. I've shown 3 of these on a human kinome scan, at the bottom here, both the early first-generation drugs, Lostortinib and midostaurin, which were multi-kinase inhibitors. And what you're seeing on the kinome scan is the selectivity for the drug for binding to any of the over 400 kinases in the human kinome. And the size of these balloons is proportional to the amount of kinase binding and generally to the inhibition of those targets. If you look at Lostortinib and midostaurin, you see that they're relatively non-selective and are multi-kinase inhibitors. The more selective inhibitor, Quizartinib, is quite selective for FLT3, which is at the top of the kinome, a little bit to the left of center, and clusters with other similarly structured receptor tyrosine kinases, such as FIMS, KIT, and PDGFR. Quizartinib is not only more selective in vitro, but it's also much more potent and is able to inhibit FLT3 tyrosine kinase more potently in vivo. And we saw fundamentally different clinical responses to the drug and actually single-agent activity to Quizartinib, which really had not been seen with the multi-kinase inhibitors. To date, the only approved multi-kinase inhibitor for FLT3 mutated AML is midostaurin, and it's only approved in combination with intensive chemotherapy. By contrast, we see single-agent anti-leukemic activity of Quizartinib in the relapsed refractory setting, where it's actually quite an active agent. But one of the problems of this drug is shown on the next slide, and that is that the durability of responses to Quizartinib as a single agent when treating FLT3 ITD mutated AML are relatively brief, as shown at the very right of this figure, where we sequenced FLT3 kinase amongst patients with FLT3 ITD mutation prior to and then at progression to Quizartinib. And what's notable is that none of these patients had additional mutations in FLT3 beside the FLT3 ITD at entry to the study, but all of them had new mutations in FLT3 that led to either impaired binding of the drug to FLT3 or would activate the kinase even in the presence of the drug. The two most commonly seen mutations were at the D835 residue, which I've mentioned previously is in the activation loop, and then another residue that functioned as a gatekeeper residue, which is similar in terms of its structure to the T315I mutation in BCR-ABL, which confers resistance to BCR-ABL inhibitors. This is shown structurally on the next slide, that these are not adjacent to each other, but actually are really in functionally different areas in the kinase. Again, the F691 residue guards the ATP binding pocket from being blocked by the small molecule inhibitor drugs, and the activation loop facilitates the kinase being in an activated state, even in the presence of the drug binding. Because these resistance mutations were so common, we got very interested in developing drugs that would be active both against FLT3 ITD and then also against these resistance mutations. And one of the first drugs that we were able to test is a drug called Gilteritinib, which we tested in a clinical trial that was called CHRYSALIS. The inhibition of the drug against FLT3 was similar to what we saw with Quizartinib in terms of it being a very potent and selective inhibitor, and in vivo inhibition of the FLT3 kinase was seen at the very lowest doses tested in patients. At 20 milligrams, we saw some inhibition of the target and some clinical responses, but both the inhibition of FLT3 kinase was more potent and more consistent at higher doses, and also response rates were higher. So we got potent and consistent inhibition at 80 milligrams and up, and that's when the responses seemed to plateau. So those are the doses that we developed going forward. We next looked to see if there were treatment-emergent mutations either in FLT3 or in other genes that occurred amongst patients treated with Gilteritinib. And what we saw were two types of mutations with some frequency. We actually did see in a few patients new mutations in FLT3, and these included mutations at that gatekeeper residue, although only in a small number of patients. And some patients actually evolved with new FLT3 ITD sequences that were not seen at the start of therapy. But much more common were mutations in either NRAS or KRAS or other genes in the RAS-MAP kinase pathway. We additionally saw mutations that would be expected to activate RAS-MAP kinase signaling, such as BCR-ABL, in patients at progression to Gilteritinib. And overall, a new sequence mutation was seen in the majority of patients treated with Gilteritinib, suggesting that, as was seen before with Quizartinib, treatment-emergent mutations that activated signaling in FLT3 or in key FLT3 downstream targets were the predominant mechanism of resistance to Gilteritinib. Now, one question that we had was, are these mutations happening in the same cells as those with the FLT3 mutation or happening in cells that were wild type for FLT3? And in order to answer this question, we had to examine the clonal architecture that was being measured. We started with bulk sequencing of patients for a panel of genes at study entry and then repeated this testing either during response or at progression, which is shown in the next slide on the right half of the panel. You can see in this subject that this patient's leukemia experienced clonal evolution with the acquisition of an NRAS G13R mutation that was not detectable at the start of therapy in, again, this bulk sampling by NGS of the patient's tumor cells. And presumably, since the variant allele frequencies of all the other mutations is holding steady, we assume that this mutation is present also in the cells that have FLT3 D835 mutation. On the next slide, we see a different pattern where we start with a cell that has an IDH2 mutation at a very low allele burden, but a FLT3 ITD mutation and an NPM1 mutation at a higher allele frequency. However, at progression to Gilteritinib, this patient effectively lost the FLT3 ITD mutation, and the dominant clones that are present have both IDH2 and NPM1 mutation, and 2 NRAS mutations are present, which presumably means there's actually 2 clones present at the time of resistance, each of them having an NRAS mutation. And here we think the leukemia has swapped cells that contain the target mutation for cells that lack the target mutation but have additional signaling mutations that lead to activation of similar targets. So this brings up an interesting question. If we're seeing clonal evolution even in cells that lack the target mutation, what is the architecture that's present? And can we really say that the acquisition of a RAS mutation is what's driving resistance, or are we just selecting for cells that lack the mutation of interest? It would be much more interesting to get higher resolution evaluation of this marrow to better understand the dynamics of how the clonal environment is actually changing in the presence of a tyrosine kinase inhibitor. And we can't really do that by using bulk sequencing. So I'm going to turn it over to my colleague, Dr. Smith, to talk about a technology with which we can resolve at a higher degree of resolution the clonal architecture in the marrow and in the peripheral blood of patients treated with Gilteritinib and other tyrosine kinase inhibitors, so we can better understand the dynamics of clonal architecture and how they change in the presence of targeted therapy of cancer.
Thank you, Sasha. So to resolve these issues of tumor heterogeneity, a potential solution is to perform sequencing of single cells. However, traditionally, single-cell sequencing has been expensive and has been low throughput due to the fact that multiple modalities were needed to be able to sort single cells and amplify the alleles of interest and to then be sequenced. Single-cell sequencing techniques have also been traditionally prone to allele dropout due to the need for whole genome amplification. And due to the need for PCR amplification, it's also been prone to PCR and sequencing errors. However, single-cell sequencing can have actual very clear advantages over bulk sequencing. In terms of variant allele frequency, usually the variant allele frequency by bulk sequencing is inferred and estimated, whereas by single-cell sequencing, you can actually quantify this. With bulk sequencing, heterozygosity is assumed, and the zygosity of the cells cannot actually be confirmed. However, while single-cell sequencing, you can actually quantify the proportion of cells that are heterozygous or homozygous mutant. As mentioned earlier, using bulk sequencing and variant allele frequencies inferred from bulk sequencing, clonal architecture can be estimated, but with single-cell sequencing, you can actually quantify and observe the actual clones that are occurring in that sample. And the way you do this is by actually being able to quantify the actual existence of mutation co-occurrence within single cells. Whereas by bulk sequencing, this is always estimated, and it's assumed that mutations that occur at the same variant allele frequency must also occur in the same cells. However, this assumption is often wrong. Using bulk sequencing, it's also impossible to estimate any contamination with non-neoplastic cells. And this needs to be done by different methods, such as by looking at the pathology specimen where the DNA was derived from. However, with single-cell sequencing, often you can actually detect the contaminating normal cells and remove them computationally by removing cells that are wild type for at all loci sequenced. In this slide, I show an attempt at single-cell sequencing the gene encoding the tyrosine kinase, that FLT3, that is often mutated in acute myeloid leukemia. In this study, we took frozen cells either from the blood or bone marrow of a relapsed FLT3 mutant AML patient and performed single-cell sorting by fluorescence-activated cell sorting. And then we performed duplex PCR in the sorted cells to amplify two regions of FLT3 that encoded the internal tandem duplication mutation, or the area of the kinase domain that is a hotspot for activating point mutations, the amino acid D835. Using this very low throughput method, we looked at 3 patients who relapsed initially on the FLT3 inhibitor Quizartinib. And on this slide, we show on the left of the circle the zygosity for the ITD mutation, in which 1 red quadrant represents heterozygosity and 2 red quadrants represents homozygosity. On the right, we show the zygosity of the drug-resistant D835 mutation. Again, 1 colored quadrant represents heterozygosity and 2 colored quadrants represents homozygosity. The relative size of the cellular population is represented by the relative size of the circle. As you can see in these 3 boxes, which each represent an individual patient, there were various cellular populations that represented both heterozygous and homozygous mixes of the ITD and drug-resistant D835 mutations. In 2 out of the 3 patients, there were also cellular populations that had either heterozygous or homozygous D835 mutations alone. And in some cases, and actually in both of these cases, these mutations were polyclonal. We also were able to note through this method that in all 3 patients, there were populations of the cells that either had heterozygous or homozygous ITD alone and no other secondary mutation within FLT3 that could explain the drug resistance of these cells. This made up 30 to 50% of the population in each of these patients. Similarly, in all 3 of these patients, we also saw a population of cells with no mutation in FLT3. These could represent contaminating normal cells or potentially non-FLT3 mutant AML blasts that were not dependent on FLT3 signaling for survival and therefore were able to survive in the presence of treatment with the FLT3 inhibitor. What this illustrates, however, is the ability of single-cell sequencing to resolve a tremendous amount of tumor heterogeneity that is present only at 2 genomic loci in a single gene. Imagine the amount of heterogeneity that could be resolved by looking at multiple disease-relevant genes in any given patient. So, to be able to resolve a larger degree of heterogeneity, we were able to utilize the Mission Bio Tapestri single-cell sequencing platform, and again, use patient samples from AML patients treated on a FLT3 inhibitor clinical trial. These were blood or bone marrow samples that were analyzed on the Tapestri platform to perform sequencing of 19 AML relevant genes at hotspots. And these samples were then sequenced on a standard Illumina sequencer, and data was processed to be able to definitively resolve the clonal composition of each of these tumor samples. We analyzed initially a total of 4 patient samples and more than 130,000 cells in total. And in doing so, we were able to resolve in each of these patients that the RAS mutations that we had newly noted at the time of relapse on the FLT3 inhibitor Gilteritinib, the RAS mutations that Dr. Perl told you about that we found by bulk tumor sequencing, we were able to identify in all of these cases that these newly acquired RAS mutations did indeed occur in the same clone as the FLT3, the pre-existing FLT3 mutation. And this is shown in these three bar plots, each which represent an individual patient. And in each of these patients, the size of the red bar represents the size of the relapse co-mutated FLT3 and NRAS mutant clone. You can see that in each of the three patients, there were also other mutations that occurred in this relapse clone. On the left, this patient had a triple mutant WT1 FLT3 NRAS mutation, made up the bulk of the relapse clone, with a smaller proportion of the blue clone, which was the ancestral WT1 FLT3 mutant only clone that was presumably still sensitive to FLT3 inhibitor. In the middle patient, you see that the relapse clone was a triple mutant FLT3 NRAS DNMT3-1 mutant clone, and you also see a small proportion of the presumably ancestral FLT3 DNMT3A only mutant clone. And then finally, in the most complicated scenario, on the right, there's a patient with multiple clones at the time of relapse. There is a red quadruple mutant, actually FLT3 NRAS SF3B1 IDH2 mutant clone, another large population of cells that is mutant only for SF3B1 and IDH2, shown here in green, that likely represents the ancestral clone, and then a small proportion of the relapse population is made up of this blue clone, presumably the TKI sensitive FLT3 mutant SF3B1 IDH2 mutant clone. What these slides show you is that the variant allele frequencies that we obtained by single cell versus bulk sequencing for each of these mutations was very close, indicating that single cell sequencing is recapitulating the findings of bulk sequencing just at a finer resolution. Again, you can see in this patient, the variant allele frequencies obtained both by bulk and single cell sequencing are virtually identical. In this slide, you can see, however, the advantage of the resolution that you can obtain by single-cell sequencing when you look at a patient over time as they are treated on Gilteritinib. On the left, you see the changes in variant allele frequency that occur over time. This patient was sequenced at 4 treatment time points, pre-treatment, 2 on-treatment time points, and at the time of relapse. And you can see that the variant allele frequency of the IDH2 and SF3B1 mutations really does not change at all. The assumption in this case would always be that these two mutations must be contained in the same clone because they are the same variant allele frequency. However, we can see with the single-cell sequencing that this is not true. You can see here that the FLT3 variant allele frequency by bulk sequencing drifts down, indicating what you would assume to be disease response. And most impressively, you can see that the drug-resistant NRAS mutation is only resolvable at the time of relapse and is not something that you can pick up at any of the pretreatment or on-treatment time points. Now we turn our attention to the single-cell sequencing and the additional resolution that you can get. These are the same time points sequenced by single-cell sequencing. And you can see at the pretreatment time point that we can clearly resolve 2 tumor clones. The bulk clone is actually an IDH2 SF3B1 FLT3 triple mutant clone, and there's a small percentage, 3.4% of the population, that is made up of a clearly ancestral IDH2 SF3B1 mutant clone only, this green clone. As you can see, during the course of FLT3 treatment, the green clone is enriched, as you might expect, for a clone that does not have a FLT3 mutation and therefore must not be driven by FLT3. It would not be expected to respond to a FLT3 inhibitor. You can see that the blue clone, the presumably FLT3 TKI sensitive clone, drops off from the time pre-treatment, on treatment, and to the time of relapse. But most impressively, you can see the drug-resistant NRAS mutant clone is actually not observable pre-treatment, but is able to be resolved very soon on treatment. At only 28 days on drug, we were able to see that 0.04% of the cell population at that time was actually made of the eventual red relapse IDH2, SF3B1, FLT3, and NRAS mutant clone. And this red clone continues to be enriched on treatment, making up 0.8% of the population after another 84 days on treatment. And then finally, after another 3 months, it makes up approximately 25% of the relapse population, with another bulk relapse population due to this non-FLT3 driven SF3B1 IDH2 only mutant clone that is clearly unresponsive to FLT3 inhibitor treatment. We performed the same longitudinal analysis with 2 other patients. And what we're able to see is very clear different patterns of resistance. On the left, you can see a situation in which the patient acquired the drug-resistant NRAS clone only at the time of disease relapse. And in fact, this clone is not detectable prior to treatment or at the on-treatment time point. In the middle situation, you can see the patient that I just explained in the last slide, in which there was a non-FLT3 dependent population detectable pretreatment, and then a early acquisition of an acquired drug-resistant NRAS mutant clone that emerged through the course of treatment. And then finally, and most excitingly, in the patient on the right, you can actually see that in this patient, the NRAS clone actually was detectable pre-treatment in a very small number of cells, and that this clone was also clearly selected for during the course of treatment at the time of relapse, made up the bulk of the relapse cell population. This pre-existing relapse clone was identified exclusively using single-cell DNA sequencing. There was no evidence of resolvable NRAS mutation at any of these time points using bulk sequencing. So in conclusion, high-throughput single-cell DNA analysis allows us to definitively resolve clonal architecture in contrast to bulk sequencing NGS methods. We were also able to identify rare, discrete, but very clinically meaningful clones. The ability to do this allows us potentially to be able to utilize single-cell sequencing for better patient stratification and for actual dynamic selection of therapy as we see drug-resistant clones emerge on treatment with targeted therapy. So at this point, I'd like to hand it off to my colleague, Dennis Eastburn from Mission Bio, who's going to tell you a little bit more about the Tapestri platform.
Hi, I'm Dennis Eastburn, and I'd like to take a few minutes to tell you about the platform that was used to generate the data you've heard about today. At Mission Bio, we've developed a technology for high-throughput single-cell DNA sequencing to better resolve heterogeneity that exists within tumor populations. To do this, we've launched commercially the Tapestri Precision Genomics platform that consists of 3 main modules: an instrument that runs disposable microfluidic cartridges, and reagents bundled in 8 reaction kits. They contain all the materials you'll need to go from a cell suspension or a nuclear isolate to a sequence-ready library. We also have developed easy-to-use bioinformatics and data analysis and visualization software that completes the solution for single-cell DNA analysis. The key product specs for Tapestri that enable broad applications across oncology are shown on the next slide. Tapestri performs accurate variant calling on up to 300 genomic loci, and it performs this analysis on 10,000 cells per sample run. This enables extraordinary sensitivity for detection of rare subclones down to or below 0.1% of the population. The platform is compatible with a variety of hematologic as well as fresh frozen solid tumor tissue samples. Mission Bio offers a number of off-the-shelf targeted sequencing panels that are available, as well as the ability for researchers to design targeted sequencing panels specific to their application. On the next slide, you can see an overview of the Tapestri workflow that's designed to fit seamlessly into existing NGS workflows that many of our customers are using. The workflow consists of first taking a cell suspension or a nuclear isolate of interest and loading that onto the microfluidic cartridge that runs on our instrument. Once this is done, barcodes can be incorporated into the amplified material from the genome, followed by downstream library preparation and next-generation sequencing. And I want to spend just a couple minutes talking about the microfluidic steps that run on our Tapestri instrument. So that's really a key differentiator in what Mission Bio does and is central to enabling our very accurate and highly sensitive variant calling on individual cells. The platform runs a novel two-step microfluidic droplet workflow that maintains compartmentalization of all cell analytes within droplets. Cells are first combined with a proprietary lysis reagent that contains a protease that not only effectively lyses individual cells, but it's able to digest nuclear proteins such as histones that would otherwise act to sequester genomic DNA and hinder efficient downstream molecular biology. Once this protease has performed its function, it's inactivated, and the droplets containing efficiently prepared genomic DNA are then combined with cell-identifying molecular barcodes, targeted gene-specific primers, and PCR reagent. At that point, the emulsion can be taken to a standard laboratory thermocycler, and barcodes get incorporated into the amplified genomic DNA. We've spent a great deal of time optimizing these droplet-based single-cell PCR reactions to afford high sensitivity, high multiplexing, and very low allelic dropout rates. On the final slide, we list a number of key application areas where Tapestri is currently being used. Of course, we support hematology applications such as the one you already heard about today. And to support these applications, we offer both an AML panel as well as an expanded myeloid panel. And we continue to develop new targeted sequencing panels for additional applications in this area. We also offer the ability to profile and resolve tumor heterogeneity in solid tumors. So we've developed a universal nuclei isolation protocol that efficiently isolates nuclei from a number of fresh frozen solid tumor tissues, such as lung, prostate, colorectal, and breast tissue. Lastly, Tapestri has been adopted by a number of investigators in the area of genome editing. These investigators are using Tapestri to accurately assess efficiency of editing as well as off-target editing events that occur within single cells. Additional information about the Tapestri platform can be found at www.missionbio.com. We hope you've enjoyed the webinar today, and thank you for your attention.
Thank you for your presentations, Dennis, Alexander, and Catherine. It is now time for the Q&A. So ask any of the speakers a question, just type it in where it says type your question here and then press submit. So our first question, and I think this one is directed to you, Catherine, and it asks, can you envision this technology being used for MRD?
Absolutely. Actually, we've already begun to explore the use of this technology in the MRD space by looking at serial patient samples from AML patients that pre-treatment at the time of remission and at the time of relapse, with the goal of seeing if the clone that emerged at the time of relapse was either detectable pre-treatment or even at the time of remission. And we've only looked at a small number of samples, but we have been able to observe in some patients that, in fact, the dominant relapse clone is actually detectable at the time of remission. And this was done pretty much using the same cell number and therefore the same sensitivity of the data that we showed in the presentation.
Excellent. Thanks, Catherine. And our next question is also for you. And it asks, in the example you walked through, could you have inferred the clonal architecture from the bulk sequencing data?
So, as we had shown actually in the slide presentation, the clones that we were able to pull out by single cell sequencing were not able to be inferred from the bulk sequencing data. In fact, based on the bulk sequencing data, you would have assumed that there was actually very little change in the clonal composition in that patient, whereas in fact by the single cell sequencing analysis, we saw a significant degree of dynamic change.
Excellent. Thanks, Catherine. And our next question is for you, Alexander, and it asks, do you think resolving clonal architecture with single-cell analysis could impact clinical decisions?
I definitely think it could impact clinical decision-making. I think the combination of mutations that we see certainly defines the likelihood of relapse with targeted agents. And here, when we're looking at least a single agent drug targeting a specific mutation, the emergence, in particular co-mutations that predict for resistance in a population otherwise targeted by a drug is a strong reason that we might envision this technology identifying relapse clones earlier in therapy than they might be present, just waiting for them to show up. And that could inform decisions as to whether to add additional agents, change agents, or consider transplantation for patients.
Excellent. Thanks, Alexander. And our next question is also for you. This one asks, with error-corrected sequencing, do you think you could have gotten sort of similar results?
That's a good question. With the caveat that I haven't personally worked with error-corrected sequencing, I would say that it's true that you could get similar results from any highly sensitive technique, but there are certain advantages to being able to show that the mutations you're looking at are actually cell-specific. For example, in one of the examples that we showed, we found an oncogenic RAS mutation in the same cells as a FLT3 mutation that was targeted by the drug. And I think when you see co-mutation, particularly for a resistance mutation, you have a little bit greater confidence that what you're looking at really is relevant for disease progression and not just a random finding. You certainly can see false positives in rare variants, and that seems less likely to be the case here. So there are certain advantages of the technique that we show.
Excellent. Thanks, Alexander. And our next question asks, what proportion of cells submitted to the pipeline resulted in usable genomic data, and what is the rate of doublet cells? Dennis, is that something you can help answer?
Yes. So we worked very hard to get our cell capture rate to 10%, and as we further develop the platform, that number will be improving. So for 100,000 cell input, we get a 10,000 cell output of cells that have usable genotype. In terms of the doublet rate, we have an extremely low rate there, and it's driven specifically by Poisson statistics. So it's performance in terms of doublets is on par with any other commercially available droplet-based platform.
Excellent. Thanks, Dennis. And just a quick reminder to our live audience, you still have time to ask any of the speakers a question. To ask one, all you need to do is just type it in where it says type your question here and then press submit. So our next question, and this one is for you, Dennis. And it asks, how is single-cell sequencing data analyzed following the workflow?
Yeah, we recognize that data analysis can be quite a challenge for single-cell sequencing, so we spent quite a bit of time developing intuitive and easy-to-use solutions, both for the bioinformatics component of data analysis, as well as a software package that we call Tapestri Insights that enables in-depth clonal analysis once you have your genotype data. And it provides a number of visualization tools also that can make a lot of sense from this high complexity data. And all of these solutions are freely available for our customers to use as part of our complete Tapestri genomics platform.
Excellent. Thanks, Dennis. And following on from that, how long does the workflow take?
Yeah, the workflow is really just an add-on to NGS workflows that our customers are currently using. And it takes about a day and a half of total time, but only 4 hours of that is hands-on time from a user.
Excellent. Thanks, Dennis. And our next question is for you, Catherine. And this one asks, can you discuss how you overcame issues with mapping of FLT3 ITDs?
So within the software package, there's actually specifically an ITD caller built in, similar to the way ITDs are called using other NGS pipelines. So, so there, in terms of the actual specifics of the programming, I'd have to defer to Dennis.
Dennis, is that something you can sort of pick up on and help out with?
That question was regarding FLT3 ITDs?
Yeah, that's correct.
So we've been able to detect 95% of all known FLT3 ITDs that we've analyzed with the platform. And we've been able to get ITDs, down in the single base range all the way up to nearly 100 bases. So we believe our, informatics pipeline and ability to call those critical ITD mutations is actually quite robust.
Great, thanks Dennis, and thanks Catherine. Our next question is for you, Dennis, and this one asks, what solid tumours have been tested on Tapestri, and how are the tissues prepared?
Yeah, that's a great question. We recognize that different types of solid tumor tissue can actually be quite challenging to disaggregate into single cell solutions or suspensions. So what we've done is actually developed a very easy to use nuclear isolation protocol that can be universally applied across different solid tumor tissues. And just some examples of solid tumor tissue that we've looked at include all the sort of major cancers that people are familiar with: breast, lung, colorectal, prostate, melanoma, as well as quite a few other tissues. And what we see is with these nuclear isolates, we're able to load them onto the cartridge and run our workflow and really obtain virtually equivalent performance from that material relative to what we see with our heme samples, such as PBMCs and bone marrow aspirates.
Great, thanks Dennis. And our next question asks, how much DNA is sequenced for targeted genomic loci? Dennis, is that something you can help with? We seem to have lost Dennis there for a second.
Oh, no.
Hi,
yeah.
Dennis.
Hi, I think I was on mute. I can answer that.
Okay.
So we offer the ability for customers to target hundreds of genomic loci, and we're constantly working on our ability to multiplex. But currently we have panels that are sequencing up to about 70 kilobases of genomic DNA.
Excellent. Thanks, Dennis. And I think this next question is also for you. And it asks, how easy is it to implement custom buffers and reagents into the Tapestri platform?
We really don't support the use of custom reagents. What we do offer is the ability for investigators to design custom targeted sequencing panels so they can provide genomic coordinates pretty much anywhere in the genome, and we have very efficiently design highly multiplexed panels, and then enable that targeted sequencing panel for an investigator-specific application.
Excellent. Thanks, Dennis. Our next question says, the Mission Biosystem seems to have a very low allelic dropout. Why is it very low? Is that something you can help answer, Dennis?
Yeah, I mean, some of that relates back to just the use of these very low volume picoliter, size droplets that we use. Enables very efficient detection of low copy, target material. And we've spent many years actually optimizing those reactions such that we have an allele dropout rate across many different loci that averages about 5 to 10%, which is better than pretty much any published single cell workflow where they've looked at that critical metric.
Excellent. Thanks, Dennis. And our next question's for you, Catherine. And it says that you mentioned the challenges related to cost throughout. And sorry, let me start again. So you mentioned during your presentation the challenges related to cost and throughput for applying or using a single cell approach. Can you comment on whether the use of Tapestri overcomes those challenges and how that might impact the ability to use this approach prospectively or even routinely based on sort of added sample processing time or cost and value?
Sure, absolutely. So, I mean, I think in an example, you know, the single cell sequencing that we showed that using the where we did the flow sorting and then targeted sequencing for FLT3 it took months to complete just for a small number of samples, whereas you know, the data that we showed you generated using Tapestri is able to be completed in a matter of days by a single user. So in terms of throughput, there's a huge advantage to the Mission Bio system. In terms of cost, I think certainly, you know, any genomic technology is associated with significant cost. I think that, but in terms of, especially if you take into account the cost of manpower, the Mission Biotechnology is, I think, significantly cost saving. I mean, it's still several $1,000s per sample if you add in the sequencing cost, but that's certainly within the window of how much it costs to do any sort of NGS sequencing assay at the current time. So I definitely think this platform has the potential to actually be practically used potentially in the clinical setting.
Excellent. Thanks, Catherine. Well, I'm afraid that's all we have time for today. Before you leave us, we would really appreciate if you can spare a few seconds to fill in a very quick survey on your experience today. I would now like to thank Dr. Catherine Smith, Dr. Alexander Perl, and Dr. Dennis Eastburn for their presentations and for answering your questions. I would also like to thank the webcast sponsors, Mission Bio, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.